The citric acid cycle is the second step in cellular respiration. It is a chain of chemical reactions that produces energy. The citric acid cycle is also known as a Krebs cycle and takes place in the mitochondrial matrix. It begins after glucose is split into two pyruvates in glycolysis and is converted to acetyl-CoA. Acetyl-CoA is a 2-carbon molecule and combines with oxaloacetate, which is a 4-carbon molecule. Together, they make citrate, which is the first step of the citric acid cycle. Now it is a 6-carbon molecule. The enzyme used for the step is citrate synthesis. In the second step, cis-aconitate is formed. This is also a 6-carbon molecule, and the enzyme aconitase is responsible for this change. Also during this step, water is released. In the third step, cis-aconitate is transformed into isocitrate, which is also a 6-carbon molecule. Aconitase is also the enzyme that makes this change. Also during this step, water enters the cycle. In the fourth step, the isocitric acid is oxidized to alpha-ketoglutarate. This is a 5-carbon molecule, and isocitrate dehydrogenase is the enzyme responsible. NAD+ is needed for this, and NADH is produced, and CO2 and a hydrogen ion is released. In the fifth step, the 5-carbon alpha-ketoglutarate is converted to succinyl-CoA. This is a 4-carbon molecule. It is converted by using the 2-oxoglutarate dehydrogenase complex. Coenzyme A and NAD+ are used. NADH is produced, and CO2 and a hydrogen ion is released. In the sixth step, succinate is formed, which is a 4-carbon molecule. The enzyme succinyl-CoA synthase is used. Coenzyme A is released, and GDP is phosphorylated to form GTP. GTP is equivalent to ATP, which is energy. In the seventh step, fumarate is formed. This is a 4-carbon molecule. The enzyme used is succinate dehydrogenase. FAD is used, and FADH2 is produced. In the eighth step, malate is produced. Malate is a 4-carbon molecule. It is formed by the enzyme fumarase. And during this step, water is used. In the ninth step, malate is converted back into oxaloacetate by malate dehydrogenase. As I mentioned earlier, oxaloacetate has 4 carbons. NAD+ is used, and NADH is produced, and a hydrogen ion is released. Oxaloacetate is also the same 4-carbon molecule that combines with acetyl-CoA to form citrate, thus completing the cycle. In the end of the cycle, for every 1 pyruvate molecule, a total of 3 NADH, 1 FADH2, and 1 GTP are produced. It is important to note that glycolysis splits glucose into 2 pyruvates. This means that the cycle goes around twice per 1 glucose molecule.